Okay, so you have your recombinant DNA, you have your plasmids, but how do you get those sections of DNA, enough of them to make these plasmids? You need to be able to replicate them, and we can't just use like cells to replicate them. So how can we replicate DNA without just growing cells, growing more and more cells? So the way that we do this is a process called polymerase chain reaction. Or PCR for short. So this is makes multiple copies of DNA fragment in a short amount of time. So what happens is you have your DNA. In this case, in this little picture, it's blue and purple. You have your DNA, and it is heated so that you, in a lab, in a specific environment, it's heated so that the hydrogen bonds are disturbed and the strands of DNA separate. Then you mix it with some deoxyribonucleotides. You add in the enzymes and the tRNA and everything necessary in order to replicate and make those two separated strands into new DNA strands themselves here. Then you just continue cycles of heating and cooling. So then those two DNA strands that you have both separate. Now you have 4 template strands, and then you can get 8 DNA strands. And so they exponentially start to grow. So what happens in genetic testing via PCR is that you can get a DNA sample, a very small DNA sample, let's say a fetal sample of, you're worried that your baby has some genetic disease. You can get a small DNA sample or small cell sample from your fetus, and then you can do PCR to replicate that DNA over and over and over so that there's enough of it for you to actually detect what you're looking for. So I'd like to go ahead and show you guys. So the main COVID-19 test, like what we do on campus all the time, is a PCR COVID-19 test. So both the nasal swab test and the spit test could be those PCR tests. So the virus does not actually have any DNA in it. They found COVID-19 particularly does not have DNA. It only has RNA. So how does this work? We just said that PCR is to replicate DNA. Well, I stole these pictures based off of a website. So the website, if you're interested to read more about it, is below. So what happens is your RNA molecule here, or the viral RNA molecule that you have, is going to, if you have COVID-19, that RNA that you have is going to be undergo reverse transcriptase. So it has this special enzyme that they put in. So this is actually something, it's from like the 80s, I think is what I read. They found a way that they can add this specific reverse transcriptase enzyme and take the RNA and make a DNA strand. So going backwards, instead of taking DNA to make RNA, they can make RNA into DNA. Then now they have a strand of RNA next to a strand of DNA, RNA-DNA hybrid. And then they're going to use an enzyme to completely break apart that RNA and just degrade it, make no use of it. So now they have a strand of DNA. They can replicate that, put a complementary strand of DNA there, and now they have DNA. Then through the PCR, they separate the DNA and they will replicate it over and over and over for a while until there's a lot of the DNA. Then what they'll do is add something called a probe molecule. So this probe here is a specific chain or specific gene, specific nucleotide chain. And if the DNA that has been replicated in this, like if the COVID-19 DNA has been, or what was RNA, but now it's DNA, is replicated, this probe will attach to that DNA chain and make a specific, it will add into that DNA chain as it's replicated. As you notice, this probe here has this little green blob. that green blob is a fluorescent molecule. So they add on to a nucleotide on this probe a part of a molecule that, when is incorporated into a DNA chain, then starts to fluoresce. So the more and more of this DNA you have, the more and more it starts fluorescing. So the DNA polymerase assembles new DNA strands from DNA building blocks. You have that primer in there, or probe in there, I'm sorry. And you just make more and more. And as you make more and more, the probes are released. So you'd make a bunch of DNA, you split them apart, put the probes on there. The probes then, as the RNA or DNA in this case, sorry, the DNA is broken apart. When this probe is broken apart, that's when it fluoresces. Okay, so when you have this the probe is torn apart, you have fluorescence or a color change. In the case for the COVID one, it is a fluorescent molecule. So it's like glows pretty much under black light. And then as you can have this building block, it gets incorporated into the molecule. So if you have, long story short, if you have COVID-19 DNA, the fluorescent molecule will start getting incorporated into the DNA in the PCR test that is incorporated. That will then code for more fluorescent DNA. And so as you do this test, more and more cycles of heating and cooling and keep replicating this DNA, you're going to get more and more glowing, or more and more indication that COVID vaccine or the COVID virus was present in that sample.